In this video, we will talk about diets and MS, and in particular about intermittent fasting. Intermittent fasting does not make recommendations about which foods to eat, nor about energy. On the other hand, and unlike the diets that we mentioned until now, it does make recommendations about the time, that is, about when we should eat. Intermittent fasting involves limiting food intake to specific periods of time and fasting the rest of the time. During the fasting, it is allowed to drink water, coffee, and drinks without calories, but not to eat solid food or drinks that contain calories. There are several patterns of intermittent fasting, and among them the most popular are time-restricted eating, consisting of fasting daily for 12 to 16 hours and eating normally the rest of the time, that is, for the remaining 12 to 8 hours. 24-hour fast once or twice a week. Or alternate day fasting, which consists of fasting 24 hours every other day. If we fast for prolonged time beyond 12 hours, as in intermittent fasting, the stored glucose and protein are consumed, as well as part of the fat. In this situation, fat mobilization is increased. Glycerol from this fat continues to make its way to the liver to participate in glucose generation, and fatty acids continue to make their way to organs and tissues that can use them as an energy source. But in addition, fatty acids also go to the liver, where they contribute to the formation of ketones that leave into the blood and go to organs and tissues that can use them as a source of energy, such as the muscle and the heart, but also the brain. This type of fasting of more than 12 hours results in reduction of fat, and therefore an anti-inflammatory effect has been attributed to it. And furthermore, ketones appear to be neuroprotective for cells in the brain, and therefore a neuroprotective effect has been attributed to prolonged fasting. If the fasting is prolonged for a long time, the reserves of glucose, amino acids, fatty acids, and glycerol can be depleted, which leads to a state known as starvation. In this state, cells cannot obtain the biomolecules they need for their functioning and survival, so they end up dying. Under these conditions, cells begin to digest parts of themselves in order to obtain the biomolecules they need to survive in a process known as autophagy. Cells can digest their own cell organelles by fusing them with vesicles called lysosomes that are loaded with enzymes. This type of autophagy is called macroautophagy. Cells can also digest other molecules in a process called microautophagy, or digest molecules, which must first bind to proteins called chaperones in order to enter lysosomes. In this case, we speak of chaperone-mediated autophagy. Once inside the lysosomes, both the organelles and the molecules are digested by the enzymes present in the lysosomes. The final result of the three types of autophagy is the generation of biomolecules that the cell can use to obtain energy or to synthesize structures that it needs for its survival. Autophagy is not only important because it allows combating starvation, but also seems to be beneficial in eliminating pathogens, in degrading damaged proteins, which are very important in neurodegenerative diseases, but also during elimination of damaged cells of tumors. Thinking about the possible beneficial role of autophagy, there are people who fast with the idea of inducing autophagy, although inducing autophagy is not so simple since we do not know in humans how long a fasting period has to last to induce it, since autophagy is intended for borderline starvation situations. In mice, it has been demonstrated that a 48-hour fasting is capable of inducing autophagy. However, it is important to note that after 48 hours of fasting, mice lose 20% of their weight, which is a lot. In fact, after 48 hours of fasting, when mice induce autophagy, they are in a precarious situation, and a few more hours of fasting would be lethal. In humans, we do not know how many days of fasting are necessary to induce autophagy. Humans only lose 2% of their weight in 4 or 5 days of fasting, which suggests that very long fasting is probably necessary to induce autophagy. More studies are required to know what type of fasting is capable of inducing autophagy in humans. Regarding the scientific evidence, there is a clinical trial that we have already seen when talking about the ketogenic diet, which was carried out with 111 people affected by relapsing remitting MS, and in which one part followed intermittent fasting for 14 hours each day, one part a 7-day intermittent fast every 6 months, and the rest a ketogenic diet. This study demonstrated the efficacy of all these diets on fatigue and quality of life. Other clinical trials that we have also seen when talking about caloric restriction diets have compared the effect of these diets with intermittent fasting. These trials have shown that for people with MS, it is easier to follow intermittent fasting than a caloric restriction diet. However, these studies could not find significant differences between groups for the parameters that had been analyzed, which were fatigue and quality of life. Based on the data we have so far, our recommendation for people with MS is that intermittent fasting is an interesting option due to its anti-inflammatory and especially neuroprotective potential. And intermittent fasting with a time-restricted eating pattern such as 14-10 or 16-8, that is, fasting for 14 or 16 hours, which can include the hours we sleep, and a period in which we can eat between 10 and 8 hours, may be a good option as it is probably easy for many people to follow. And it can be combined with a healthy diet such as the Mediterranean diet in such a way that it can add to the benefits of a healthy diet.